Hello everyone, I welcome you all once again to my series of lecture that is Understanding Pharmaceutical Science with Dr. A.J. Hariharan. So today we are going to understand about the microbial culture media. So in this lecture we will first understand what is meant by a culture media, what are the characteristics of the culture media, and we will discuss elaborately about the different types of culture media. So first we will understand what is a culture media. So a microbial culture medium is maybe a solid or liquid preparation generally used for the purpose to grow the microorganism in a laboratory or to transport the microbial culture from a collection center to other laboratories or from between any two laboratories, and also to store the microorganism. So this culture media will be generally used for almost all the purpose for studying the microbiological works, right? So what is the basic need of a culture media? So if you want to study about a particular microorganism, the first thing what we have to do is we have to grow the microorganism in the laboratory, and also we have to maintain the morphological and biochemical integrity of the microorganism. So there we need this microbial culture. So this culture media will provide necessary nutrients for the growth as well as maintenance of this microorganism in a laboratory. So a suitable culture media is needed, especially to study the microbiology of that particular microorganism, right? So for doing this, first we have to understand that what is the basic characteristics a culture medium should have. So an effective culture medium must contains all the nutrients necessary for the growth of the microorganism. That means the microbial culture media should carries all the necessary nutrients which is needed for the growth of the organism. So the nutritional requirement of the organism I have already given in a separate lecture. You can see through a video lecture. So the major nutrients, that is the major source of nutrients which is needed for the growth of the microorganism in the form of a carbon source or an energy source or a nitrogen, phosphorus or a mineral source, which should be added into this culture media for its growth. So if you see the various characteristics of this culture media, it generally includes that we have to favor the growth of the bacteria, both in terms of the chemical constituents as well as in terms of the hydrogen ion concentration, pH of the media. For example, if you see some culture media will favor the growth of a bacteria but doesn't favor the growth of the fungi. Basically, even the pH of the media will do this activity. Generally, bacteria has an ability to grow in a pH of the culture media from around 7 to up to 10 or 11, but the fungi can grow in a pH of the culture media less than 6, around 4 to 6, right? So the characteristics of the culture media should be not in the nutritive components itself, it also favor the hydrogen ion concentration also. So our job is to properly select or design an appropriate culture medium for cultivating in the laboratory. So to do this, we need a complete knowledge about the microorganism normal habitat. So generally different microorganism will grow in different natural conditions. So some grow in a very cold places like a psychrophilic bacteria which is having different chemical environment, and some bacteria can grow in very hot springs like Thermus aquaticus, which can grow at a temperature of around more than 80 degrees Celsius like a thermophile, and its nutrient composition will be different. So to select or to design an appropriate culture medium, we need a knowledge about its natural habitat. So based on this natural habitat, we can design or we can select an appropriate culture medium. So these natural surroundings will help to constitute what are the nutritive requirements of that particular bacteria or a fungi or any type of microorganism, so that we can design a precise composition of the microbial culture media. So each and every microorganism will have different nutritional requirement. So the precise composition will depend upon each and every species. So a satisfactory culture medium will need a precise composition which can vary from one species to another species, both in terms of chemical constitutions as well as the pH. Right. So let us understand what are the different types of culture media are there. So we can classify the different types of culture media basically on three basic backgrounds. One, based on its physical state; second, based on its chemical composition; and third, based on the function. We can call this a functional types of media. So the first classification, we can classify the media, microbial media, based on its physical state. So physical state itself, it can exist in any different types of physical states. Based on it, we can classify into three types. One is the liquid state the media can present. Second is the semi-solid state the media can present, and the solid state. Generally, the media present in liquid state we call as a broth culture, and if it is in solid state, generally we regard as an agar culture. The reason is that the physical state of the media can be changed for the same nutritional composition only by adding a particular solidifying agent. Major type what we use is the agar. So in liquid or the broth culture media, there is no presence of agar will be there, nil agar will be there. In semi-solid, around 0.5 to 1% of agar is added, whereas in solid state we generally add 1 to 2 percentage. The most optimal condition is 1.5% of agar is added. So this agar changes the physical state of the media. For example, consider a nutritive media, nutritional broth. So a bacteria, we have a common called nutrient broth, so which carries all the nutrients and it exists in physical state as liquid state. And nutrient agar, it carries all the nutrients at the same composition, but 1.5% of agar is added, so it becomes a solid media. So the important part by differentiating this physical state is the solidifying agent agar. So the agar is generally used as a solidifying agent. So it is a polymer made up of D-galactose, 3,6-anhydro-L-lactose and D-glucuronic acid. So this polymer is generally ideal in nature. It doesn't interfere with any of the microbial biochemical pathways, and also it is non-toxic to the bacteria. So it is commonly obtained from red algae. So it is isolated from red algae and it is widely used in all the microbiological laboratories as a solidifying agent. Apart from agar, we can use gelatin also as a solidifying agent. Other polymers are also available, but it is not satisfactory as that of agar, because there are various reasons why we use agar as a solidifying agent. Is that the one of the major reason is its solidifying ability. Generally, agar can melt at around 90 degrees Celsius. Once we cool down until 45 degrees Celsius, it is in liquid state. After that, it starts solidifying once it reaches the room temperature. So this temperature up to 45 degrees Celsius, so it is in liquid condition, it will be helpful for handling the preparation of the nutrient agar plates, because the temperature is well tolerated by our hands, so we can pour into the petri dish. As well as if we are using a pour plate type of culture, we can add the microbes at this temperature, because the microorganism also doesn't kill at the temperature. So this is the major reason why you use agar as a solidifying agent. Second, once we grow a microorganism in its agar medium, we can grow at different levels of temperature, because the microorganism may be a psychrophile, mesophile or thermophile, whatever it may be. Different temperature of incubation can be done with the agar medium without affecting its physical state. It will be in solid state only. And moreover, agar is an excellent hardening agent, because most of the microbial can grow in the agar without degrading it, because it doesn't have any enzymes which has an ability to degrade this agar medium. So the agar has been widely used as a solidifying agent in the microbial media, especially to convert the physical state from broth to a solid state. Right. So the second classification of the microbial media is based on its chemical composition. So based on chemical composition, we can classify into two types. One is the defined media or a synthetic medium. Second is the complex media. So the name defined media itself tells that the all the chemical components what we are adding in this media will be defined in nature. That means if I adding glucose 1 gram means glucose molecular formula is C6H12O6. In 1 gram, I know how many elements of carbon I have added, how many elements of hydrogen I has been added or oxygen is added. So we know the well defined components, what are all we are adding in this media, right? So this defined media is generally used for various purposes, especially an industrial microbiologist. We need a defined media to understand the particular characteristics of the product formation. But commonly it is used for the photolithotrophic autotrophic bacteria like cyanobacterial cultivation and certain protistic photosynthetic protist cultivation. This defined media is preferred. So it is mainly used in many purposes. The major purpose of usage in the research is to study the metabolic process. So if I add this carbon source, what is the metabolic output? If I change this carbon source from glucose to a maltose, what is the metabolic output? If I use a starch, whether the microorganism is producing amylase or not. Like that we can identify by using the defined media, right? For example, a medium of a E. coli, you can see in the table. So a defined media of E. coli needs a glucose 1 gram, disodium hydrogen phosphate 16.4 gram, potassium dihydrogen phosphate 1.5 gram, ammonium sulfate 2 gram, magnesium sulfate 200 milligram, calcium chloride 10 milligram, and ferrous sulfate 0.5 milligram in a liter of deionized distilled water. So the media is prepared and sterilized around 121 degrees Celsius for 15 minutes and the pH is adjusted to 6.8 to 7 as a defined media for the cultivation of the E. coli, right? So the second type of media is the complex media. The name itself tells us this media is little bit complex in nature because we doesn't know the exact quantity of each and every element present in the media, and moreover the chemical composition of unknown in nature. So this becomes a complex nature of the media. That's why we call it as a complex media. So the complex media, if you see, it generally carries certain chemical like peptone, beef extract, meat extract, or yeast extract. It doesn't have a well-defined chemical constituents, right? So, for example, consider a complex media like nutrient broth media or nutrient agar media, which is a complex media generally composed of a peptone and yeast extract. But the use of this complex media is very important because we can use this complex media in wide areas. So this complex media will have all the nutrients which can support the growth of any types of microorganism, right? So if we put E. coli, it will grow. If we put pseudomonas, it's also will grow. Like that, all the microorganism can sufficiently grow in this complex media. So we can use this once we doesn't know what is the exact nutritional requirement of the bacteria. If we don't know, we can cultivate into this complex media, that is the nutrient broth or nutrient agar in the case of a bacteria, right? And the third type of classification is that we can classify the media based on the functional. So functional type of media. So there are different types of functional types of media are available. Some media can be used for sterility tests, like that it can be there. But majorly we can classify into three major groups. One is the general purpose media or supportive media. Second is the enriched media, or sometimes we call as enrichment media, and third is the selective media, right? So let's understand one by one. First is the general purpose media. So it is nothing but it supports the growth of all types of microorganism. That means it is not like a complex media. It has a defined nutrients, but still it can support the growth of many organisms. For example, tryptic soy agar or citric soy broth can grow many types of fastidious bacteria, right? Second is the enrichment media or enriched media. The name itself tells it is enriched. That means it carries an additional nutrients for the growth of the microorganism. So we additionally add some blood or other special nutrients like cholesterol to the general purpose media so that it can favor the growth of some fastidious microbes. So an example is that this enrichment media is that blood agar is an enriched media. It is otherwise called a specifically fortified media or specially fortified media, right? So the last one is the selective media. So the name itself says the selective media. That means it selectively favors the growth of a particular organism. For example, if we want to grow only the gram negative bacteria and we doesn't want the growth of gram positive bacteria, means to that media we can add certain dyes or bile salts like a methylene blue or crystal violet or basic fusion. If you add into the media, it will inhibit the growth of the gram positive bacteria, but it favors the growth of the gram negative bacteria. Thereby we can do the identification. For example, Indo agar or yes using methylene blue agar or mechanical agar is generally used to identify E. coli in some substances. So for example, if you have a municipal water corporation, I want to know whether this carries E. coli or it is less than the particular limit. So this can be identified by cultivating into this agar. If E. coli is there, it will favor its growth, but it will inhibit the other bacterial growth. So the E. coli can get nutrients and it can grow comfortably. So this is called as a selective media. So thank you very much. I think this lecture will be very helpful for basic understanding about the microbial culture media. So thank you very much. So continuously view my various video lectures. Thank you.